Hi students, welcome back to our YouTube channel. As we were doing the chapter of biochemistry that was amino acids and protein, and today we are going to discuss the part 4 of that chapter, and today the topic is properties of amino acids. Alright, so first of all we have to categorize the properties into two categories. The first one is physical properties and the second one is chemical properties. Okay, and you know that the chemical properties considered as like the reactions, while the physical properties, okay, we are going to discuss here the physical properties like solubility, melting point, taste, optical properties, ampholites, Zwitter ions, and isoelectric pH. So first of all, you have to remember for the physical properties, like in case of solubility, so you have to remember that the amino acids are soluble in, most of the amino acids are soluble in water. Okay, so most of the amino acids are soluble in water and insoluble in organic solvents. And the next one, melting point. So remember that the amino acids mostly melt above 200 degrees Celsius. So it means that the melting point is very high. Okay, and next one, the taste. Remember that according to taste, like the amino acids are categorized into three like divisions. The first one is like sweet, the second one is tasteless, and the third one, that is like bitter. Okay, so remember that like amino acids are divided into all three divisions. Like for example, some of the amino acids may be sweet, like glycine. Here I am going to write glycine, alanine, okay, and valine as well. While the tasteless, that are like for example here I am going to write that is leucine. While some of the amino acids that are bitter, these are arginine, okay, and isoleucine. Okay, so that's why they are used as a flavoring or a food flavoring, or they're used as a flavoring agent in the food industries. Okay, and next one is optical properties. As you know that the amino acids contain like a carbon that have four different groups. For example, here is hydrogen, here is NH2, here is carboxylic acid group, and here is R group. Okay, so it means that mostly the amino acids are optically active. Okay, because of the four different groups with the carbon, okay, central carbon, except the one that was, or that is glycine, because in the glycine instead of R here is hydrogen is present. Okay, so that is in case of glycine. So glycine is not optically active, while the other all the like amino acids that are optically active. Okay, so that's why the structure like they are present as a D isomers and L isomers. Okay, and next one is amino acids as ampholites. Number 5, amino acids are ampholites. Ampholites means that amino acids contain both groups, like carboxylic group as well as NH2 group. Okay, so they can donate a proton, or accept a proton, hence they are regarded as ampholites. Okay, so actually the Zwitter ion is also like a part of the ampholites. So here we will discuss in detail about how they donate proton and how they accept proton. First of all, you have to remember that what is meant by Zwitter ion. Zwitter, the name Zwitter is derived from a German word which means hybrid. Okay, Zwitter means hybrid. It means that the like any compound that having the both type of properties, like here in this case you have to say like both type of ions, positive ions and as well as negative ions. Okay, so how is it? Because the amino acids really like rarely exist in the neutral form. Okay, so in strongly pH the amino acid is positively charged cation, which while in strongly alkaline pH or high pH in alkaline it is negatively charged anion. Okay, so each amino acids has a characteristics of pH, like leucine that is exist as like you can say like the pH 6 at which it carries both positive and negative charges, and that exists as a Zwitter ion. For example, here in this case Zwitter ion will be look like that. Okay, so here you can see the negative group and positive group that are present on like a same compound. Okay, and next one is isoelectric pH. Isoelectric pH is defined as, actually that is also like a part of Zwitter ion. So in the isoelectric pH you have to remember that the pH at which the molecule exists as a Zwitter ion, or dipole ion. Okay, and that carries no net charges. Like for example, the pH value can be calculated by taking the average pKa values. Like for example, here we have to calculate the pH of by taking the average pKa values of both as like negative ion and as well as positive ion. So here I am going to write pK1 of COO negative, and the second one pK2 that is NH3 positive here, and divided by 2. Okay, so you can calculate it, that Zwitter ions, by this formula. So actually here the ionizable groups, actually corresponding the ionizable groups, like for example in case of leucine has two ionizable groups. So it means the pKa value or pH of the leucine can be calculated from this. How? By, for example, here I am going to show you, like that is a neutral one. Okay, and when the hydrogen is donated, so it means that is now converted into, for example here the hydrogen ion is donated, so it means that is converted into RCH. Now here it will be NH3 positive. Okay, while in this case you can see here there is a cation formation and like a positive charge cation formation, so that will exist as low pH. Okay, and in case of like loss of electron, for example here the hydrogen is expelled out from the group, so here it will be considered as RCH NH2, and in this case there will be a negative charge appear on carboxylate ion, so it means here that will present as an anionic form, so it means that is present at high pH. Okay, so now from this you have to make a Zwitter ion by, here in this case there is the loss of hydrogen, and in this case there is the gain of hydrogen, where the hydrogen will be gained at this group NH2 group, and hydrogen will be lost from the carboxylate ion. So here it will make like R CH COO negative, and here it will be NH3 positive. Okay, so that is known as Zwitter ion. Okay, and in this case you can calculate the Zwitter ions, like the pH of the Zwitter ion. So here, as I said, the leucine exists as the like pH 6. So how it will be, like for example here pH of pKa first of carboxylate ion plus pK2 of NH3 ion divided by 2. So here it will be, the pH will be pKa of carboxylate ion, that will be 2.4, plus pK2 that were NH3 group, that is 9.6 divided by 2, that will equal to 6.0. So that is the pH for the leucine. Okay, so these are the physical properties of amino acids. Okay, now we are going to start the next one, and that is chemical properties of amino acids. So in the chemical properties, as I discussed earlier, that the chemical properties consist of the reactions. Okay, so remember that there are the two types of the reactions because of carboxylic group or carboxylic acid group. Like for example, amino acids form salts, that will make salts, or that can make esters. How? Like as you know that the salts that consist, like for example here replacement of hydrogen, and here is the addition of sodium, so it means that is making of like a salt. So it means when it react with the bases. So here I am going to write how, in which condition that will make salts, when it will react with bases. Okay, here is form salts when it react with the bases. Okay, and it will form esters like COOR when it react with the alcohols. Okay, and next one is like reaction with ammonia. So amino acids can be react, or the carboxylic group or carboxyl group of the dicarboxylic amino acids react with the ammonia to form amides. So here it will form amides when it react with the ammonia. Okay, like in case of aspartic acid, like aspartic acid react with NH3 to form like asparagine. Okay, so it means amino acids when react with the ammonia to form amides normally. Okay, and same like in case of glutamic acid, that will react with the ammonia to make glutamine. And the third reaction, or the third chemical property, that can amino acids can be react with the, or that have ability for the decarboxylation. Like decarboxylation means amino acids undergo decarboxylation in which to produce what? Amines. by the loss of what? By the loss of carbon dioxide. Okay, like in this case I am going to write a general reaction. For example, here is RCH and here is like NH2 that will undergo decarboxylation, and here is it will make RCH2 NH3 positive. Okay, so that will make like amine. So here in this case there is the loss of carbon dioxide take place. And the fourth one, that is like amino acids can be react or due to ninhydrin reactions, or amino acids can be react with the ninhydrin. So in this case the alpha amino acids react with the ninhydrin to form purple like purple, blue or pink color complex reactions. Okay, like here I'm going to write amino acids like AA react with ninhydrin to form, like here in this case it will make keto acids. Okay, along with elimination of ammonia, and along with here is carbon dioxide also evolve. Okay, so and here the next byproduct that will be like hydrogenate. Okay, and same here the hydrogenate that will undergo again like you can say addition of ammonia here in the hydrogenate, and along with the ninhydrin, and that will make a Rohmann's purple. Okay, so that is the amino acids like due to ninhydrin. Okay, sorry, here add number 5, that is number 5. Okay, number 4, that is amino acids due to NH2 group. NH2 group is actually like you can say amino acids behave as bases and combine with acids to form salts. Okay, so here it will be